Hello and welcome back to the MCAT grind. We have Sensation and Perception from Psychology Chapter 2. So let's get right into it. Sensation is defined as stimuli, so anything that your five senses picks up, right? Like color, or you feel this touch, or you smell a particular odor. Those are all sensation, like direct stimuli from the environment. So if I see something in the distance that's like a black and white blob, that's what the sensation is. It picks up that exact stimuli. Now perception though, it is your brain making sense of that stimuli. When that sense travels up to your brain and you get all these nerves processing these stimuli, you get a definition of what that stimuli is. So that black and white blob from a distance, from what I picked up visually, it has been processed by my brain to be a cow. That's what it was, right? So making sense of that stimuli and attaching meaning to it. Now, in sensation, there's an absolute threshold, so the minimum amount of stimuli to trigger your brain to notice, right? So my hand could be like not touching anything, but then all of a sudden there's a little bit of wind blowing. When the wind blows a little bit, and I feel that that's the minimum amount of sense I can feel. If there's just like some buzzing of a fly next to my finger, I might not be able to feel it. I need something more to trigger feeling that sense. Now, in response to that, we also have a relative threshold, and specifically in Weber's law. Weber's law says that to tell a difference between two stimuli, let's say put your hand in water, to know the temperature, your body, you sense a difference of the exact proportions. For instance, knowing whether it's 10 degrees versus 12 degrees, you feel there's a difference between the water temperature. Or when you stick your hand in a 100 degrees Celsius bathtub versus sticking into a 120 degrees Celsius bathtub, then you can feel a difference. If it's below that, let's say 10 degrees versus 11 degrees, you don't feel a difference. Okay, so the just noticeable difference is set in a constant proportion, right? This time is like 20% proportion, right? It could vary from person to person, but it's very set for a specific sense. Okay, we also have detection theory, saying that whether you get successfully say there is a signal and detect it is yes, and if you miss it, then it's just a miss, right? Let's say I'm closing my eyes and all of a sudden I feel like there's an alarm buzz nearby. I wake up and I look towards my phone and nothing, it didn't go off. So that is a false positive. When there is no signal, I wake up and thinking there was, right? That's a false positive. Correct negative is when I successfully just stay asleep because I know that there's no alarm going off. Right, and going on to what we see, there are Gestalt principles. These are what we determine to help us group things a lot easier. Right, we are lazy creatures and we like to make things simple for us. So looking at this, the starting first one, we have continuity. Continuity means that we determine this to be a square kind of pattern plus a wiggly pattern instead of grouping this part and this part together, right? In closure, we can say that instead of three little wiggly worms like this, we say that it's like a triangle altogether because we mentally can close this part together in a closed shape. Much easier for us to draw and recall. This one is proximity. We can group these three little dots, boom, 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 together, right, into their own little groups because, well, it's just easier for us to group them. We, instead of counting each one, we go like, oh, there's three groups of little dots. Contour means that from this shape, you can kind of tell that there is a square in the middle, even though these are four different various shapes. We can say that, okay, we see an obvious square shape in between them because of the shape that is not there, but it makes it there. It's very similar to closure, but closure, there's a very specific points. attaching and tracing the outline to. Alright, and also the triangles are basically here. They are kind of forming the ends of the triangle, whereas these are just flat lines and edges. Similarity just means that we want to group the triangles together and the dots together as it goes. Alright, we have our touch senses. We have our nociceptor, which feels pain. Noci means pain, and we just feel pain from those. And these are all present on our skin. I believe they're below the first layer. I think they're into like the deeper epithelial layers. That's where these receptors are found. Merkel cells, you can think about murky deep water, deep pressure. Also, when you feel and put your hand in like the deep muddy water, you can feel that there's a grippy muddy texture. Merkel cells. Meissner's corpuscles are responsible for feeling light touch. You can think of Meissner, maestro. Maestro is a conductor, and they need to be very gentle in conducting the orchestra. In Pacinian corpuscles, they're responsible for deep pressure as well, but they're also for vibration. I don't really have a good recall for this one. I think Pacinian, maybe Pac-Man, right? Pacinian, when you chomp, and then you eat what's it called, the berry, and you suddenly vibrate and you become invincible. Maybe. We have free nerve ending, and they're very painful if you pull on those. There's been a time where there's been experiments, unruly experiments, and trying to pull on the free nerve endings of people, and that was excruciating. I don't recall, well, I do recall which events those are from, but free nerve endings also for pain and temperature. Ruffini cells or corpuscles are for stretch, feelings of stretch. So you can feel when you stretch out your skin, that's your Ruffini receptors feeling that stretch on your skin. Okay, for taste, we have sweet, sour, salty, bitter, and umami. Sweet, we know what it is. It's sugar. Sour, you can feel that lemon in your mouth. Salty is feeling that chips or maybe really salty ham. Bitter, you can think of eating eggplant or what else is bitter? Brussels sprouts. Yeah, that's kind of bitter. Umami is special because it was a lot later found to be that savory taste you get after eating a really oily dish. I guess that triggers a lot of glutamate excitation in your brain, and it gets addicting also. So those are your five senses, and they're not exactly distributed perfectly on your taste bud, right? Before, there was an actual map showing like they're only found in specific regions, but it has been found to be proven false. They're found pretty equally distributed all over on your tongue, and you have a lot more near the tip. Okay. We also have our smell that travels from olfactory receptors to the bulb. So smell is one of the most fascinating senses, because when you pick up all these different odors into your olfactory receptors, they go straight to the olfactory bulb and go up to the higher level processing centers in your brain. What that means is it skips the thalamus. Remember, thalamus was one of the organization's switchboard centers in your brain that helps relay sensory information. By skipping over that, they help trigger memories a lot better, because they're basically odors in their purest form. So we can know thousands and thousands of different smells, right? So that's it for sensory and perceptions, and let's get right into the visual and hearing. For visual, you have your eye, and it has multiple parts. Starting from the outside, I cut out a lot of other sides and made sure that there's only ones that you need. Cornea is the outermost part, like the hard part. You can think of it as just a hard part outside of your eye, and that protects and shields from any debris. You have your white part, sclera, right? That's basically the white of your eye, the majority of the outside what we can see in your eyeball. To control the pupil, the size of the pupil, we have your constrictor pupillae that can constrict. Do you remember which nervous system is under control of that? That's right. Constricting pupil is parasympathetic control to relax, and when you are under stress, you want to dilate to take in as many sensory information as you can get. Your lens help focus the light onto your retina, and your choroid lines with muscles that you need to help support the eye. This entire eyeball is filled with vitreous humor. I don't think that is a big part of the MCAT, so the other parts have been cut out. Fovea is a specific place on your retina with lots of cones. Cones are divided into red, green, and blue. So cones help with color, and rods help with shades of black and white, shades of dark. So rods work really well in like darker times where you can see black and white imaging, and cones are really well with color, red, green, and blue. Okay. So what we can see here from a top-down view, we have your left eye and your right eye. You can see that everything in your left visual field from your left eye, right, as well as the left visual field of your right eye, go to your left brain. Why? Because the middle part, close to your nose, crosses to the optic chiasm, right, with the middle visual field. They cross at the optic chiasm and cross to the respective brain side. So what can we say? We have two visual fields, your left visual field and your right visual field in each eye. Left visual field all goes towards the left side. Your right visual field all goes towards the right side, no matter what eye it is. So it goes from the retina and then travel through the optic nerve, cross through the chiasm. If it's in the middle visual field, it will go through the lateral geniculate nucleus, which is the LGN in the thalamus, finally going to the visual cortex in the occipital lobe for processing. Now for hearing. Hearing, we have the pinna, which is like the soft part outside of your ear together with your earlobe. That's where the sound can be caught and conducted really can be caught like a fishing net and traveling through your outer ear canal. Once it comes to the tympanic membrane or your eardrum, it vibrates, and so that it transmits and conducts the sound into like a little vibration. It hits the malleus like a hammer. It doesn't really look like this, but it's just for better visualization. Hammer, and an incus is just the think about this is a hammer. Incus is like the little thing, stick, and stay piece is the thing you're hitting on. So boom, boom, boom. Malleus, incus, stay piece. And when you hit stay piece, the stay piece will then transmit through the oval window the sound all the way through the cochlea, which looks like a snail. When the sound gets traveled through, the inside fluid called the endolymph moves around and then exit out the vibration through the round window. The round window then goes through outside to the vestibular cochlear nerve and then goes to the medial geniculate nucleus in the thalamus, finally going into the audio cortex in the temporal lobe. So for both of these, vision and hearing, they're probably the highest tested in the sensory parts of the MCAT. The two additional info to know is the superior and the inferior colliculi, as we saw in the last chapter. They're for the video and audio reflex. So if you suddenly hear a loud sound, you turn your ear and your eye would focus on the source. And that's pretty much it for sensory perception. See you later.